Hello, everyone. My name is Brianna Ankney, and I'm the Senior Inside Sales Account Manager for Los Angeles and Silicon Valley. I am delighted to welcome you all to today's webinar, Sequencing the Wild, Unlocking Animal and Marine Metagenomes with Oxford Nanopore. It is my pleasure to introduce today's speaker. Kobun Truelove is a research specialist in the Biological Oceanography group at MBARI, where he studies how and why marine ecosystems change over space and time. His work focuses on developing environmental omic methods to improve the detection of marine biodiversity to help transform how life in ocean is monitored and managed. He holds a PhD in environmental biology from the University of Manchester, a master's in fisheries science from Oregon State University, and a bachelor's in cell and molecular biology from University of Washington. Prior to joining MBARI, he completed postdoctoral fellowships at Stanford University's Hopkins Marine Station and the Smithsonian's National Museum of Natural History. Ariel Rabines is a postdoctoral scholar in the Integrative Oceanography Division at Scripps Institution of Oceanography. He holds a PhD and master's in marine biology from the University of California, San Diego, and a bachelor's in molecular cell and developmental biology from the University of California, Santa Cruz. His research focuses on microbial community dynamics, real-time environmental monitoring using nanopore sequencing, and ecological genomics. He has extensive experience in molecular methods, bioinformatics, and field-based oceanographic research. Dr. Rabines has collaborated on numerous publications exploring microbial interactions in coastal and open ocean systems, and has contributed to projects aimed at improving agile cultivation for sustainable biofuel production. Candace Williams is a technical service manager at Oxford Nanopore Technologies, where she leads the Western United States field application team. She earned her PhD in molecular biology from Mississippi State University using next-generation sequencing and analytical chemistry to study interactions between the gut microbiome and dietary shifts in both giant and red pandas. Prior to joining Oxford Nanopore, she led the microbiome projects at San Diego Zoo Wildlife Alliance, where her work focused on how microbiota played key roles in managing healthy species in both captivity and the wild. She also developed microbial-based treatments and methods to sequence samples anywhere, while training the next generation of conservation-focused microbiome scientists. Following the presentations, the speakers will be available for question and answer sessions. Please add your questions in the Q&A tab. You can also upvote any questions. Now, Candace will start us off with an introduction to nanopore sequencing technology. Thank you all for joining us today.
Thank you so much, Brianna, for the introduction. My name is Candace Williams, and I'm the Technical Services Manager for the Western United States. I'm so excited to be speaking with you all today as a part of the Sequencing in the Wild webinar. Before we get to our two fantastic speakers, we want to provide a quick introduction to nanopore sequencing for those of you who might be new to the technology. As our name suggests, nanopores lie at the heart of our technology. We use nanopores to sequence DNA or RNA by passing these molecules through the nanopore using a motor enzyme. This allows us to directly sequence both DNA and RNA, preserving rich epigenetic information without the need of any additional library prep. We can also read any length from 20 bases up to 4.3 bases on a single platform. We can sequence in real time. So as these molecules are streaming through the pore, they can be base called, and that data can be accessed by the user before the run even completes. It's also quite modular, so you don't need to wait until you have enough samples to run. You can run whenever you're ready. We can do that because we have different scales at both the flow cell and device level. For our flow cells, we have our minion flow cells here on the bottom left. This is our lower throughput flow cell that generates about 20 gigabases of data. We have here on our right our larger throughput, our higher throughput flow cell, our Promethean flow cell that generates about 100 gigabases or more potentially depending on the application. We have different sizes of our devices. For example, we have our handheld, as you can see here, our Mark 1D, quite small, perfect for field-based applications. We also have our larger benchtop devices, like our GridION, that can sequence 5 minion flow cells in parallel, and our Promethean 2 series that can sequence 2 Promethean flow cells in parallel. We also have our larger devices, like our Promethean 24, that can accommodate up to 24 flow cells in a single run. So depending on what the question you have or what your goal is, we have a device that can suit your needs. Our portability for our smaller devices really allows us to sequence pretty much anywhere. Our devices have been deployed across so many different diverse and extreme locations. Today, we're going to hear about 2 different projects involving marine settings. However, you can see we've been almost everywhere, including space, which is really exciting. In order to be able to sequence in any of these locations, you first have to prepare your libraries. We're going to quickly talk about some options for library prep. We've already touched on briefly sequencing options with different scales of our devices, whether they be more suited for field or lab-based applications. And we'll touch on briefly some analysis portion, including an example with our epitome workflows. We have so many different types of library prep kits, right? So depending on what your experimental needs are, we have a kit to match those. From both RNA and DNA, we have recommendations. I'm not going to read this entire table to you, so don't worry. I just want to point out that we have different library prep kits depending on what your needs are. Do you need more output? Is it rapid turnaround time required? Are you going after ultra long reads, or do you have low input? These are the kinds of questions that we can answer with different library prep kits. If you want more information, please check out this QR code down here. You can find out more about our library prep kits for both DNA and RNA. Focusing more on our DNA-based approaches, especially thinking about metagenomics and in the environmental DNA context, we have some different suggestions for you. We have 2 types of kits. We have our ligation-based kits and our transposon-based kits. While they're fundamentally different in terms of how they attach the adapters onto the end of the molecules, there are some similarities between these different kit types. For example, for both kit types, we have both singleplex options and multiplex options. For our multiplex options, you can multiplex up to 24 or 96 samples, depending on the kit that you choose. You can also preserve that base modification information, regardless of whether you use the ligation or transposon based. Really, the key differences here relate to time. So if you need rapid turnaround time, the transposon-based kit might be the best fit for you. You can see for the single sample, you can go from sample to sequencing, or genomic DNA to sequencing in about 10 minutes. That's about an hour for our ligation-based kit. Still pretty quick though. The other consideration is whether you want to preserve longer fragments. So if you want to preserve the full length of your molecule, the ligation-based kit might be more suited for your needs. The transposon-based does cut those, so your fragment, you will continue to fragment your DNA. However, our ligation-based kits will then just add adapters to the end of your molecules. So if you're looking at mapping to a database, for example, for a metagenomic approach, either of these kits would be suitable. But if you're interested in assembling genomes, for example, metagenome assembled genomes, our ligation-based kits would be beneficial to preserve those longer fragments. However, if you're doing more targeted based approach, like traditional metabarcoding approaches, we do have some solutions for those as well. The first one I want to point out is our PCR barcoding kit. So this is a more traditional 2-step PCR based approach where you would do your locus specific PCR with tailed primers. You would then take those and then do a second PCR using the barcoded primers. Once all your products are barcoded, you could then pull those together and put those in our ligation sequencing kit. So this is a more traditional 2-step PCR that you might be familiar with in terms of metabarcoding approaches. However, we also have another option. So we have our microbial amplicon barcoding kit that was just released. So similarly, you do this locus-specific PCR. Our kit comes with 16S, so that would be V1, V9, so full-length 16S, and ITS, so that would cover ITS1 to ITS2 primers. But you can also use custom primers. Now, for metabarcoding approaches, you might be interested in, things like 18S, 12S, OX1, for example. You can use custom primers. However, I do want to caveat that it would be up to the individual user to validate those primers with the use of this kit. We have only validated the primers that come with this kit here. But I think we'll hear a little bit more on this kit from Ariel later in terms of the use of using custom primers with 18S. Really quickly, once you have your locus-specific PCR completed, you can then really quickly add your amplicon barcodes and then move forward with sequencing with about an hour's time. So pretty quick. And if you want more information about this kit, please check out this QR code here on the bottom right as well. One thing I want to highlight from both of these approaches is you're not limited by the length of fragment you can sequence. You're really limited by the length of fragment you can PCR. So for both of these, in comparison to more traditional metabarcoding approaches, using maybe the V4 region, for example, for 16S, using the full length 16S will allow you to get enhanced microbial community profiling with high taxonomic resolution, which is really important to be able to distinguish between closely related taxa. So both of these approaches, you can have greater taxonomic resolution by increasing the fragment sizes or these amplicons sizes that you're sequencing. And if you too want to generate a nice Sankey plot like this for your microbiome, you can. So we have some support for that as well. We have our epitome workflows. So I put in the top here, you can see the web address for that. So please feel free to check this out. You can see the workflows that are available and even downloadable data sets. So you can see here recently we've added data sets for both 16S amplicon and ITS amplicon. So if you want to test those out virtually, so before you even do library prep, you can test this out on your own with just data analysis, you can. But what you can do is you can pull these different workflows. And for example, we're going to talk about one workflow in particular, but there are a couple that might be beneficial, like workflow 16S and also workflow metagenomics for these types of applications. So we're going to highlight just this one here with workflow 16S. And although it's called workflow 16S, it can accommodate both 18S and ITS as well. So you can take your base call data from Minnow and then put that into the epitome workflow, so workflow 16S. What this will do is it'll generate this quick real-time read classification using Kraken 2, but also do more fine-scale classification with Minimap 2 plus databases. What you'll get from this is a nice intuitive report, including those plots like I just shared, and then also reports with classified and unclassified, even alpha diversity plots. So all this is available within this workflow. If you want more information, again, please check out the QR code there. But what I hope I've demonstrated to you today, even though we're looking at this from a super high level, is that we have solutions for both library prep, devices for sequencing at different scales, as well as analysis options. And while this was a very high level overview, I'd be really happy to take any questions that you might have in the Q&A. But without further ado, I'd like to kick off our next speaker, Kobun. So I'll let him take it away. Thank you.
Thank you for the introduction. My name is Kobun Truelove. I'm a research specialist at the Monterey Bay Aquarium Research Institute, also known as MBARI. Did my PhD at the University of Manchester in the United Kingdom and a postdoc at the Smithsonian National Museum of Natural History and also Stanford University's Hopkins Marine Station. And for both of my postdocs, I was working on methods for nanopore sequencing. And here at MBARI, I'm doing something very similar, working on nanopore methods for sequencing environmental DNA while at sea. And that's going to be the topic of this seminar today. So briefly, marine environmental eDNA is a way that we've been using to monitor species in the ocean without necessarily having to see them. And it gives us a way to really target larger areas of ocean ecosystems and deeper waters as well that have been, possible previously. And how we go about that is, the animals are shedding DNA either through their tissue, metabolic waste, skin and scales. And when that gets into the environment, it can be filtered out using, and we tend to use 0.2 micron filters. So we'll be collecting anywhere between 1 and 5 liters of water and then putting it through a fine filter, and that filter will collect the particulate, and within that particulate is where we're extracting the DNA. And then you really have two different methods that you could use. You can either go to a species specific primer and run quantitative PCR, or you could go directly to sequencing that DNA, and you can either sequence it with targeted genes, such as 12S, CO1, 18S, and 16S. These are kind of some of the classic targets that will hit different taxonomic groups ranging up from bacteria up to fish and marine mammals. And there's also PCR-free approaches that go away from targeting any specific gene and just directly sequencing all fragments of DNA that you extract. And so in this talk, I'm going to go over both of those methods. So I'm going to go over a targeted gene approach and also a metagenomic approach where you're just directly sequencing whatever fragment of DNA gets extracted. So fortunate here at MBARI, just showing us our location. We're located right off the coast of Central California in the Monterey Bay, and we're right next to an enormous marine canyon that is roughly the size and depth of the Grand Canyon. And we have pretty much a brand new research vessel that has recently arrived. And for this talk, I'm going to go over how we use nanopore sequencing on board this vessel to sequence eDNA and kind of speed it up so we could get results back within a day to kind of inform us while we were on the research cruise. So I'm just going to show you this video. This is basically the area that we were surveying. This is the harbor next to where we're located. This is our new ship, the research vessel David Packard. And as the talk goes on, I'll go over some of the capabilities of this ship. And you can see right below is our research institute off to the left, now the right. And our research cruise was just directly offshore there, as you can see. And we only have to go about 15 minutes offshore to get into that deep canyon area. And for this talk, I'll go through the types of diversity we found in the canyon using environmental DNA. And so we brought a nanopore sequencer on board the ship, and this was the area that we mainly surveyed. So this site here where it says C1, that's one of our long-term monitoring sites. It's right at kind of at the mouth of the canyon, very easy to access. And we were quite interested in looking at how fish migrate up and down from the depths of the canyon during the day, and then at night when darkness falls, coming up to the surface, and then as sunlight returns, they move back down into the depths. And this is something that's happening all over the oceans of the planet. It's kind of known as one of the world's largest migrations. It's really happening every day. So this is something we've been studying for a long time, and we wanted to see, can environmental DNA help pick up this signal and also provide us more information on the species as well that are migrating up and down the canyon. And so there are ways to spot where the fish are located using sonar. So we have sonar on the ship, and these red areas, and I'll kind of zoom in on those more on the next slide, are kind of areas where there are aggregations of fish. So here's kind of a zoom in of that sonar. And for example, we could sample, directly having this information directly within these aggregations. And so how we did this was using the CTD capabilities of our research vessel, and we have a 24-bottle Niskin rosette that could be lowered to the precise depths that we're interested in surveying. And those bottles, once they hit the depth, will snap shut, collect water, and then that can be brought back up to the surface. And from there, it can then be filtered, and we can work on the eDNA extractions and sequencing on board the ship. And one of the big breakthroughs that's been made here at MBARI that has really helped streamline this whole process of sequencing eDNA at sea is our third generation environmental sample processor known as the 3G ESP. So we brought this on board. And what this allows us to do is filter um, one to, well, really as many liters as you want, as long as you don't clog the filter. But we were essentially filtering 1 to 5 liters of water. And then the particulate that was caught in the filter could also be extracted using a lysin go enzyme. So it basically takes a little over an hour to filter the water, and then it's a one-step lysis extraction using the enzyme. And then that extracted lysate is then pushed through that filter again to filter out any cellular debris during the lysis process. And then it can go straight, that extraction lysate can then go straight to either PCR or directly to metagenomic sequencing. And we were able to do this with a relatively small amount of equipment on the ship. So I had a mini thermal cycler for running PCRs for the targeted primers, and then also a magnetic block for bead cleanup and library prep for the nanopore sequencing, and of course the nanopore sequencer and a centrifuge. But this was relatively light amounts of equipment. And this is something that I worked on previously for my postdocs and also over the years at MBARI, really trying to streamline this process to make it as simple as possible for sequencing at sea. And so now we're able to really get it down to, this is something it could be done once or even twice a day with minimal crew of 1 to 2 people going through all the lab work and sequencing. And if you want to do the metagenomics, the amount of time is even quicker because you don't have to do the PCR step. The data analysis afterwards is a little more complex. And for that, we're fortunate that our research vessel has a high-speed internet connection, and we were able to send all of the data onto the Chan-Zuckerberg ID pipeline, which is a metagenomics pipeline that can then go through all of the data cleaning and bioinformatics over a large cloud network that then allows um, your your sample to be analyzed and getting taxonomic data back in roughly about 20 minutes. So that saves quite a bit of time, and it's very nice to not have to do all of that computational work on board the ship, and having the internet connection to kind of send that off to Chan Zuckerberg ID really helped us quite a bit. So here's just a overview of our lab. So it's a relatively small space. We wanted to have an area that was away from where all the main work of the ship was doing to try to minimize contamination. So we had a separate room, and you can see I just have laptop, mini thermocycler, and also our 3G ESP environmental sample processor for filtering and extracting the DNA. And this is just me going through some of the lab work. And this is also very grateful for the help of Nadia Loff, who she helped build and design the 3G ESP, and she was also helping operate the 3G ESP for doing all of the sample filtration and DNA extractions. And so once that DNA was extracted, I would either go through and do PCR or go through and do the metagenomic work. And so what did we find going back to C1, which is our site right next to the opening of the Monterey Canyon? And these are just some of the highlights. So we were out there for a total of 5 days, but the first day and the second day were quite short days. We kind of arrived late and left early, which really kind of left 3 full days for eDNA sequencing. And so some of the quick highlights using this method were white sharks, tuna, sea lion. We also found hake and halibut, but essentially the big take-home message is that this is something that we can now do on board ships, to get eDNA data back within 1 day to help guide research on board large vessels, And it really helped us quite a bit to have this automated filtration and extraction system. This could also be done by hand as well, but for processing lots of samples, it really does help to have automation. So what did we find on day one? Let me kind of break this down into something that's a little easier to see than this species tree. So essentially, we found on the first day, we got good hits for bat rays, Pacific Dover sole, lingcod, rockfish, hake, anchovy. And that was all from one sample collected at night right off of that canyon site. We did the same thing on the second day, also collecting a nighttime sample. And you can see we've also found some different species that we didn't find the first night. So we found sand dab, some California halibut, pygmy poacher, again, rockfish and lingcod and hake were showing up. And we found Pacific herring, anchovy, and California sea lion. And as well as picking up a species of seabird, the common murre. On the third day, I tried the metagenomics approach. And this is something we're still working through all the data that's come back. So I'm just giving you some quick highlights off of this approach. But we found a long read for tuna, around 2,886 base pairs that match very nicely to one of the chromosomes on the tuna genome. We also found a 717 base pair read that aligned nicely to a white shark messenger RNA. And what was also quite interesting as a side story to this research, while we were out at sea, we were also very fortunate to be working with another group at MBARI that operates what's called the Piscivore cam, which is a camera that's mounted onto an autonomous vehicle. And this autonomous vehicle was patrolling the area a little offshore of where we were at C1. And this is some video off of the Piscivore camera where it also detected tuna and white sharks. And so we're feeling optimistic about these data, but there's a lot of work that we're still trying to optimize with the PCR-free approach. So while it's much easier to work with in the lab, interpreting the data is much more challenging than interpreting the data with the PCR-based approach that targets known gene markers. And so just going to briefly go on to some of our future research. So one of the next steps we'd like to do is, while we have now optimized the environmental DNA capabilities for our research vessel, we'd also like to try to miniaturize and automate that process for use along our fleet of autonomous underwater vehicles. And so this will really help us in the long term kind of scale things up to cover much larger geographic areas of the ocean. And our first objective is really trying to get the nanopore sequencer embedded into one of our long range autonomous underwater vehicles. And so we have vehicles that we've been using for several years now that already have environmental DNA sampling capabilities with a larger 3G ESP. So it's the same technology that we brought on the boat. It's a larger version that can sample up to 60 environmental DNA samples. It can also extract the DNA. So the next step is really kind of automating the lab work that was done on the ship. So this is something that could be done on the vehicle. And this will really open up a lot of exciting opportunities for research in the future for doing adaptive eDNA sampling. For example, knowing where these fish aggregation hotspots are, you could send the vehicle to specific locations to collect samples. And we can also start integrating the environmental DNA with other suites of autonomous vehicles to give us a much more holistic picture and idea of what's happening in our oceans and give us a better understanding of how things are changing over time. So I would really like to thank everyone involved in this research, in the Biological Oceanography Lab, the MBARI ESP team, the LRAUV team, the PySaborCam team, and also thank Todd Walsh for the beautiful video he gave us of the RV David Packard. And I'm very grateful for everyone here listening to this seminar. And if you're interested in learning more about the research that I do or have any questions about environmental DNA or sequencing at sea, please reach out to my email address. Also, if you're interested in following MBARI or any of their social media platforms or getting their newsletter, please follow that QR code. And I'd also just like to express my sincere gratitude for everyone who has kind of helped me along the way of this process of trying to understand environmental DNA sequencing more and the power that it has to help us understand how the world's oceans are changing. And on that note, I would like to pass this on to our next speaker. Thank you all very much.
Thank you. Happy to be here. My name is Ariel Rabines. Today I'll be talking about how we are utilizing nanopore sequencing for adaptive ecosystem monitoring in commercial algae farms and Antarctic fjords. I'm going to give some background on how I got started with this project. It was this type of work, and that was during my PhD, working with Lisa Ziegler on the Productivity Enhanced Algae Toolkit, or PEAK Challenge. That was a DOE funding initiative to optimize growth in high lipid open air algae ponds. So we were fortunate to collaborate with Global Algae on this project at their commercial operation outside of Hui, Kauai, as seen on the previous slide and right here. One goal of this project was to develop methods and tools to identify potential pathogens and beneficial microbiome components using next generation sequencing. So these cultures are started in the lab as inoculum for small outdoor raceways, which are eventually used for inoculum larger and larger raceways. They grow 2 elite strains, a green algae and a Nichia diatom. And so we were interested in seeing how these communities evolve through time, but also how different nitrogen sources affect the community composition, as well as isolating different aspects of the community. We wanted to look at the phycosphere associated bacteria, that is, organisms that are living, very close to or on the cell wall of our elite algae strains. And so the goal of this project, was to develop a rapid detection system for some of these pro or pathogenic microbes, and to use sort of the Illumina sequencing data that we generated to create probes for this SpinDX instrument that was developed by Sandia National Laboratories, who were also collaborators on this project. And unfortunately, the SpinDX system wasn't sensitive enough, and we couldn't really get it to work for this system. But we did get some really interesting data out of this project, and we ended up utilizing it for something down the line here. But really quick, I just want to share some of the data we generated from this project. So here is a Bray Curtis NMDS plot of the 16S V4 V5 data for both strains plotted together. We see that each strain has very unique, distinct communities associated with different algal strain, different nitrogen communities, but also between the phycosphere, attached community and free living planktonic communities. We also wanted to look at, long-term grow outs and see which sort of microbes are associated with productivity and low productivity. So we had 2 sort of long grow outs in July and October of 2018, and we had a crash in July 2018 and a successful grow out in October of 2018, and we're able to identify taxa that were differentially abundant between those 2 grow outs and sort of create targets for our probes for potentially pathogenic and beneficial probiotic taxa. Like I said, the SpinDX didn't work, but through this project, we were inspired to put in another DOE funding call, and that was the Algal Productivity Exceeding Expectations program. So that started in 2022, and the goal here was to use nanopore sequencing to do rapid detection of pro and harmful bacteria for these ponds. So we developed rapid library prep and data processing pipelines on site at their farm in Kauai. So this was all done in a trailer on their farm. So you can see us, we were able to collect samples, process, extract nucleic acids, prep libraries, and do sequencing all within less than 12 hours. sometimes more, depending on sort of what platform we're using, what kit we're using. So for this project, we're utilizing the MK1B nanopore device, using the off-the-shelf 16S and rapid barcoding kits. Um, this was early in 2018, so we were still using the R9 flow cells, and the quality of the sequencing wasn't quite where we'd like it to be, but things have rapidly been changing. And now with R10 and the Q20, we've seen great improvements in that. We also developed a custom two-step 18S pipeline for them. So, while Nanopore had tools that allow us to observe the bacterial and total community, we wanted to hone in on the 18S or eukaryotic community of these ponds. So there is just the algal elite strains are in there, but you also have ciliates, other grazers that could potentially be detrimental to your grow out. So we wanted to create pipelines that would capture total community, eukaryotics, prokaryotics, and viruses. And so that two-step 18S protocol really kind of allowed us to look at what sort of other protists were in these ponds. We would annotate that with like a PR2 protocol, a custom pipeline that we handed off to them as well. And the goal of this project was not only to like observe, how these communities shift with different conditions, right? So light, air, temperature, nutrients in ponds, but also these contained photobioreactors, but also to develop these tools and hand them off to these cultivators, but other cultivators in the future, and really create a robust near real-time monitoring tool so that when these cultivators are growing ponds or growouts that are seemingly do well, we might be able to catch some of these like really harmful detrimental pathogens early on and either come up with mitigation strategies, so maybe some antibiotic or probiotic treatment to these ponds, or cutting our losses before they invest more money into these grow outs. So if we can tell these cultivators, hey, we've picked up this so-and-so bug, chances are this grow is going to be unsuccessful. They don't have to put more resources and money into this, and it's a win-win. So this was a really fun project. Unfortunately, funding for this got put on hold in 2025 and is indefinitely on hold. But we were able to utilize the methodologies that we developed here, and apply them to other systems. And that sort of leads me to the next project. But I also just want to highlight really quick how well Global Algae sort of took these methodologies and ran with them and integrated them into their regular workflow. So John Patterson, Josh Brown, sort of the scientists at JAI, really sort of dove into this and were really excited by it, optimizing their workflows, for their particular needs. We come from the more environmental, marine, oceanography background, and so they work on these farms every day. They were able to kind of like optimize what we'd given them even more, reducing some of the time with extraction and things like that. For their uses. And these are some figures that were sent to me from our collaborators at Global Algae. So they were doing regular monitoring, sample collection and monitoring, and using not just our pipeline, but also epitome analysis software to just, monitor their ponds. And I'm working on a publication right now on that project. We have seen that, comparing to Illumina sequencing results from previous years, that we're getting closer and closer as the quality of these nanopore reads increases. And yeah, I mean, it was a viable method for them, and they were really happy with the results. But unfortunately, we're not working on that project anymore. But I am going to talk about how we use these resources for a different project, and that's the Genomics at Sea program. So this is a really cool, unique project. It's a collaboration between Viking Expedition Cruises, Scripps Institution of Oceanography, and the J. Craig Venture Institute. The GASP is dedicated to understanding oceanic phytoplankton in the Southern Ocean. Phytoplankton are the world's lungs. They're the base of the food web, and Viking has essentially converted their ship's COVID laboratories into a molecular lab, and started these really cool collaborations with researchers all over the world. We essentially have a research grant from Viking Expedition Cruises. This pictured here is Christian Johnson. He's a third-year PhD student in our lab now, and Jack Flowers, one of the scientists that is part of the expedition team on Viking Cruises. So Viking is sponsoring Christian's PhD. They're giving us a research grant, and we are fully integrating into their expedition team for the Antarctic season, part of the Great Lakes season. And you might be asking, like, what is Viking getting out of this? It's really guest engagement. And so Viking expedition cruises are these more intimate cruises. They're not thousands of people guests. It's closer to like 250 or 300, which is still large, but they're able to go into these really exotic places like Antarctica, like the Arctic, like Patagonia, and do these expeditions. So guests are getting out on these zodiacs, they're landing on shore, they're exploring the natural environment, and the type of guests that go on these cruises are excited by the research that we do. And so we do guest lectures, guest engagement, we set up workshops on board, and we get really, really great access to this incredible site that's otherwise really difficult to access. And Nanopore is getting all this cool guest engagement and interactions from us. So we are utilizing Oxford Nanopore sequencing to provide critical insight into how this environment is changing, from seasonality viewpoints, but also climate stressors and anthropogenic changes. So we're not just collecting genomic samples. Obviously, we need to collect metadata. We have Jack here with the CTD that's going to collect salinity, temperature, conductivity. I don't know exactly what Christian's holding here, but some sort of other metadata device, I imagine. And you have Viking Octantis in the background here with some glacier. So beautiful place to be. I got to go in the end of 2024 as a corporate guest, sort of getting this off the ground. And we just wrapped up our full last season of 2025 and started our next one. So, why are we so interested in the Southern Ocean? Well, like I said, phytoplankton form the base of the marine food web. They play a key role in global carbon cycling. And in places like the Southern Ocean, they're incredibly dynamic and sensitive to environmental changes. So we're asking questions like, how does the composition and function of phytoplankton communities change from place to place, week to week, or year to year? And another question we're looking at is like how penguin colonies, through biogenic inputs of iron, might be impacting phytoplankton communities near these penguin rookeries, and looking at the impact of them. So because the MinION is this portable device, we're able to bring the sequencing capabilities on board the ship, generate data while we're still in the field, and potentially use that for feedback on where we want to sample next, where we want to return to, and it also gets around another limitation of working in Antarctica, and that's the logistics of moving samples from Antarctica back to where you want them to be. So instead of shipping lots of samples on dry ice, we're just shipping back a few small boxes with DNA samples. Obviously, there's other samples that we're collecting, like nutrient samples, that we need to ship back or get from the ship when it comes stateside, and there's some delays there. But getting this initial data while we're down there is incredibly valuable. So this is some data from our initial sequencing run in 2024. This is using the two-step 18S, ONT overhang primer method with a MinION MK1B and a Flongle, flow cells. The Flongle really worked well for us in this application as we were doing, small pools every week. We didn't need, all that crazy sequencing depth that we'd get with the MinION. And our initial sequencing runs were really successful. We could clearly see, different microbial communities associated, or phytoplankton communities associated with different sites. Here at Des Moines Point, we saw a community is dominated by dinoflagellates, whereas in Brown Station, we saw majority diatoms. So really fun trip for me getting to go as a corporate guest, but also helping set up the molecular lab and developing some of these methodologies on the ship was really fun. And we manned we integrated into the expedition team for the entire 24-25 season. We even had some individuals go up for the part of the Great Lakes cruises for some training. So Emma, Christian, Lisa, and Bryce, all members of our lab, rotated down as, guest scientists in the expedition, as part of the expedition team, collecting these samples, doing the sequencing, and having a great time. Here's Emma at one of the first ice landing that any of these Viking expedition ships had done. So they got to walk on the sea ice. Really incredible stuff. Here's the sites that we visited for the 24-25 season. Many of these sites got visited twice. So this is the Western Antarctic Peninsula. The tip of South America is just north of here. So the cruises embark from Ushuaia, which is the southernmost city in Argentina, do a 10-day trip around the Western Antarctic Peninsula, landing on some of the islands and the main continent before going back and swapping out guests. And so, we have our team members on there for multiple cruises, getting to visit sites multiple times. Here's some pictures from that season and some of the guest engagement and sample collection that we were doing. So GASP is sort of like the next evolution in another project that is taking place down there called the Fjord Fido, and that's a project that one of our postdocs, Alison Cusick, has worked on really a lot over the last decade. So here she is, and those projects are happening in coordination with each other a lot, and sample collections happening together a lot, but they are distinct. Fjord Fido is more about guest engagement, where with GASP, we're really trying to build this really large, robust metagenomic data set, but they complement each other really well. So we have Allison, Jason, Jack, Emma, out in the field. Here's Emma back on the ship doing some large volume filtration. And here are some of the results from this first year. Again, this is utilizing our two-step 18S protocol with the MinION fungal platform. We developed our sort of own in-house analysis pipeline called PullShift. So once our team member ran a sequencing run, they would just basically point to where the data was in PullShift, and this pipeline would run using the PR2 database. So immediate sequencing and analysis, generating some taxonomic bar plots. And we did have some issues, on some of these sequencing runs. I'm not sure if it was, issues with some of our reagents staying good through the whole season, or some of the flow cells that we're using. Obviously the fungals have been discontinued, and I can kind of see why that is. We've had some really, really good runs, but also struggled with others, where we receive them at the same time. Zooming in on one of these runs that are focused on some of the larger penguin colonies, we saw some interesting results right away. We do see this interesting shift where when you're closer to the penguin colony and far away, we see minor shifts in the colony in the composition of phytoplankton. But also the greatest variation is between sites, and so sites that don't have rookeries and do, we see a lot of variation between what phytoplankton are dominating there. So this is really fun to be able to bring these figures into our lectures and say, hey, remember that penguin colony that we were just at? This is the phytoplankton that were in the water there, and then we might even have a workshop with the microscopes where they get to investigate as well. So that was a great season. We are still processing some of the environmental samples that we got as far as nutrients and metabolites. So, and we also are processing all the samples that we ran on nanopore sequencing in the field with Illumina sequencing for some validity there. And we just started the 25-26 season. Christian, who's not me, he made this calendar. He just left. I spoke to him on the phone this morning. He is on the Viking Polaris now, on his way through Drake's Passage towards the Western Antarctic Peninsula. He's got his everything he needs to do, sequencing at sea. He'll be there for 4 legs, and we'll swap out for a master's student in our lab, and then actual undergraduate who will be taking over from Caitlin. We have some other visiting scientists who will be down there. I will not be participating this year. Last year, I actually got to go not to Antarctica, but I was on the Patagonia leg going up, wrapping up some of the sequencing and prepping for this coming up year. And I know I'm going a little bit over, but I really want to quickly talk about the next evolution in our 18S protocol. So Nanopore just released this microbial barcoding kit, and we kind of wrote it off because it seemed like it was geared towards 16S and ITS, and we were really trying to develop a one-step 18S protocol. We were looking at designing sort of inline barcoded primers to use with the ligation protocol. But when I spoke to Candace recently, she told me, hey, this kit could potentially be used with any barcodes. There's nothing, they don't need to be special. There's no overhangs. And that got us really excited. So we quickly got a kit and we've been testing it out over the last week. And I just want to share some of those initial results. So this is sort of a taxonomic relative abundance bar plots for 18S, comparing our one-step versus two-step PCR method from 3 different sites from Antarctica. And we do see that we're capturing the same communities, and we're actually really pleased with the results from this microbial amplicon kit using regular primers. There's no overhang. It makes the PCR optimization much easier, and we're not having to order super large crazy primers. So looking at this, we looked at, this isn't stats, this is just really initial analysis. We see that we have much better diversity using the single step PCR compared to our 2-step method. However, we're still capturing the same communities. And so we're really happy with this. It seems to be faster, cheaper. Our read qualities are significantly better. We're like almost 50% of our reads are above Q20, better diversity. And we're starting to test this out with other marker genes, including CO1 and others as well. So, next steps, we're wrapping up the Illumina sequencing analysis to compare that to the 24-25 season, processing our nutrient metabolite data, and testing out new marker genes with this protocol. And also been talking to some collaborators at JCVI about using this methodology for some clinical diagnostics, potentially for human health, because JCVI's got a mix of different researchers there. So with that, I just want to say thank you to all the collaborators and people who worked on this project with me. It's been a real pleasure going to these amazing sites and doing field work in Hawaii, Antarctica, Patagonia. So thanks to everyone at JCVI, Scripps Institution of Oceanography, Viking, Global Algae, and DOE and Nanopore for having me here today.
I'm afraid that's all the time we have for today. Thank you once again to our speakers, Ariel, Kobun, Candace, and thank you all for joining us today. You can find more information related to today's broadcast using our QR code on the screen. If you do have further questions about your projects or getting started with Nanopore, please email us at support@nanoporetech.com or your local representatives. Thank you all once again, and I hope you enjoy the rest of your day.